All right, it just started. So this is GM2 presentation for the project called Cool Beans, and it is me talking, Marwa Buthim, with Grace Hart and Dev. All right, slide one. A moment. The coffee industry is a very big and lucrative business that, as you see on the map, it encompasses so many developing countries and hence make a good portion of their revenues. However, due to the climate change, droughts, and water scarcity, the coffee industry is threatened and these countries are facing significant losses.
Due to climate change, 60% of all types of coffee plants are at risk of extinction, including Coffea Arabica, which is the coffee species we will be working with. Furthermore, fungal growth on leaves of the coffee plant has also caused a 40% decrease in coffee production over the past 20 years. So, based off these two facts, we can see just how endangered coffee is around the world.
There are also global water shortages, which are occurring in countries where coffee is the main cash crop. This can all be solved by using the GTL1 gene, which comes from Arabidopsis thaliana in the mouse ear cress. The expression of GTL1 gene in the coffee plant will actually repress SDD1, which regulates stomatal density, and this will help solve the water shortage because water will need less plants to cool themselves off and will be better suited for dry environments. This hasn't been done before, so it's very innovative and original. And this
In this portion of the presentation, I will give a quick overview of what we will go through. First, we will isolate and amplify the DNA sequence of target gene GTL1. Second, we will produce a recombinant vector. Third, we will select and confirm that what we have is a genetically modified C.Arabicae plant. And last, we will be measuring the success of our procedures.
So the first step is to isolate the GTL1 gene. In order to do this, the Arabidopsis thaliana will be purchased from Millipore Sigma, a distributor. First, the cells will be homogenized, and then mRNA will be isolated and extracted from the cells. Afterwards, reverse transcriptase will be used along with the following primers to make a cDNA library of the GTL1 gene. Also, to amplify the GTL1 gene, PCR techniques will be used with the same primers except with T's instead of U's in order to account for DNA.
For step 2, we will be producing a recombinant plasmid. This will be done by first using agrobacterium tumefaciens binary vector PBID19. The PBID19 plasmid will be purchased from the life science market in Massachusetts, United States. Then we will use the restriction enzyme HIND3 and XBAI to cut up the PBID19 plasmid. The enzyme XBA1 will be purchased from Thermo Fisher, Michigan, United States. The enzyme HIND3 will be purchased from New England Biolabs, Massachusetts, United States. Then we will insert the GTL1 PCR product into the PBID19 plasmid using DNA ligase, and recombinant plasmid is needed to insert gene into bacteria and have it replicated.
Now we will transform agrobacterium tumefaciens with the recombinant plasmid using heat shock. Then select the transformed agrobacterium. We will grow them in kanamycin. Next, we will break open the bacteria and extract the DNA using the appropriate techniques. Then using a probe with the GTL1 DNA sequence, we will mark the GTL1 gene and isolate it. The picture in the middle shows the steps that we will use in the lab to make a genetically modified Arabica plant, and we will let you read on your own, which you will find it contains all the necessary steps, from treating the agrobacterium to transforming them, then co-cultivating them with the plant spits, and selecting the success.
The final step is to measure the overall success of the project. First, we can use the Western blot technique to search for the GTL1 transcription factor proteins. If these proteins are found, we can confidently say that the gene was inserted into the coffee's DNA and is properly being expressed. If no proteins are found, however, we can run a PCR test along with gel electrophoresis to see if the gene is present in the coffee's DNA. And if the gene is determined to be present, then there's a problem in expressing the gene, and further tests can be run to find out why there's a problem in expressing the gene. If the gene is determined not to be found in the coffee's DNA, however, then we can run further trials to properly insert the gene into the coffee's DNA. Lastly, an experiment will be run with control group of normal coffee plants versus a group of the modified coffee plants. Both will be put into a dry environment and they will be grown. Overall, the modified plant should have greater rates of success and should be able to live longer due to its less need for water.
We do need grace. What is grace?
What happened?
I don't see grace around. I'm gonna pause. Okay.
So this is our vector diagram. It is made from the PBIN19 plasmid mentioned earlier in the method section of the presentation. And the GTL1 gene, the coding sequence is the orange part that is under the larger gray insertion of the vector diagram. And the HIN3 is, as you can see on the table, the restriction enzyme that we used. So is the XBA1, and other important enzyme markers are also on this function table.
Now I'm gonna be talking about the ethics that's come with modifying the coffee plants. Potential ethical issue would be that people may oppose consuming coffee from genetically modified coffee plants in fear it may have negative health effects related to it. And to solve this, we can test whether the GM coffee plants have negative health side effects by conducting tests and experiments on animals and humans for these guidelines. Another field would be that making GM is complicated and expensive, which is very important because this research was made to solve the developing countries' issues. So we can make infographics that are easy to understand in all languages and have small workshops to educate small farmers. And for the potential environmental issues, there is the risk of contamination through cross-pollination or water contamination. We can test whether algae plants will cause water contamination or have negative effects. Another risk is risk of gene flow from the genetically modified plants to native plants, and it can be solved by developing crops with physical barriers that separate the populations.
Looking at the evolutionary context of the GTL1 gene, we can determine how our project would be fairly successful. We have constructed a phylogenetic tree for the family Brassicacea, which the Arabidopsis thaliana belongs to. Looking at other plant organisms related to Arabidopsis thaliana, they have also found success in these dry environments. And this increases our project's likelihood of success, as other similar organisms with similar genes have already demonstrated that they have an enhanced ability to survive in lower water conditions. Furthermore, Arabidopsis thaliana and Coffea arabica are in the same class, and so we can expect the insertion of the GTL1 gene into the Coffea arabica genome to be pretty successful. Looking at possible evolutionary consequences, however, the modified Coffea arabica would have a differential reproductive success among wild plants, and so if this plant was released into the wild, it is possible that it could take over the ecosystem due to its lack of dependence on water.
There are many directions that our project could go. Some ideas are, in order to combat a hesitant customer base, we could monitor the interactions with the environment in order to prove that a genetically modified coffee plant is a good idea all around. It'll protect the water shortage, it'll protect future farmers For a second idea, we could analyze how genetically modified coffee plants affect native species through gene flow. This is more of an idea to protect the future direction that coffee is going. We obviously don't want all of our coffee to be only genetically modified. We would still like for some natural plants to be growing. A third idea is we will find out if there is another cheaper method in order to protect the farmers who are at risk of buying a more expensive bean. And this will reduce the costs for the consumer, for the farmer. It's a great idea. The last final future direction that we might have, we will study whether the gene insertion in other plants will make them drought resistant as well. This could be applied to so many other cash crops in order to develop economically struggling countries.
So thank you for listening to our presentation and watching our presentation. And so these are references that we use throughout the presentation.
All right. Cool. Well,
Any questions? No?